record the session. Okay, so I'll record the session.
Yes, sir. So you can share for us.
So is it visible?
Yes, visible, sir.
Okay, Nilanjan, anything? Any query?
No, sir, it is by mistake. Sorry, sir.
Okay, so very good morning, students. Today we will start the formal introduction to the course of bioinformatics. In our previous class, we had a generic discussion about the course, like how we normally perceive the subject of bioinformatics from the computer science point of view, and what we are going to study, and more or less what are the scopes of the subject. And we also saw a short video, and how, like, the central dogma of molecular biology, how it works from the gene, from the process of transcription to the process of translation, and how from the chromosomes, the unfolded DNA strand, we make a copy of the necessary or the relevant segment in the DNA strand, and we call it as a gene, and then make a copy of the gene, and that messenger RNA goes out of the nucleus. And before going out of the nucleus, there are some splicing process, that cutting some of the irrelevant parts, and after that splicing, it goes out of the nucleus, and it enters into the cytoplasm, and there you have the protein factory, that is the ribosome, that actually reads the messenger RNA. Three codes, or we call it codons, three nucleotides at a time. Okay, in the DNA and the messenger RNA, we call it as a nucleotides, and three nucleotides at a time, and then form a finally a chain of amino acids, and those amino acids essentially form a protein. And even after that is the process of translation, and after the translation, you can see that a mature protein forms after post-translational modifications, and they are these proteins are essentially the building blocks of a living organism, and are responsible for almost all the essential activities in any living organism. So this process is called the central dogma of molecular biology, and almost all the activities that we do, starting from like studying the genome, that is the study of genomics, then the transcriptomes, like the messenger RNA, how they carry the copy of the gene to the protein factory, that is the transcriptomics, to the conversion of messenger RNA into a protein, that is the study of proteomics. So all these together we uh, generally call it the molecular biology. And then the computational aspect of this molecular biology leads to this fascinating subject that is called bioinformatics. That is like biology and informatics. So when we apply informatics, the science of informatics science, into biology, we call it bioinformatics. So this is the general overview that I gave in the last class, and today formally we will start again with the introduction to the subject, and we'll see how and what are the overall scope and spread of the subject. And in the subsequent classes we'll actually elaborate one specific area that we'll discuss in this introductory class, and see more specific algorithms or more specific details that are relevant to this particular study. Again, keep in mind that bioinformatics that we are studying here is we are studying this, and we have formulated the syllabus in the form of a elective subject in a Masters of Computer Science and Engineering course. Okay, so this is essentially a computer science course, and we would like to see like how we can actually solve the biological problems using computational algorithms. So you can expect that there will be a lot of algorithms. You will have to solve several numerical problems, and also you'll have to study, like there may be a, you can say 50-50 or 60-40 more bias towards computational segment, and little bit of biology, of course, you'll have to study, but nothing serious. And if you have not studied biology beforehand, then also I can assure you that you can pretty well understand the terminologies. And if you miss out somewhere, please interrupt me, and you can always ask questions in the chat box. And also you have access to me through WhatsApp and other phone calls. You can always talk to me and clarify on any specific topic. Okay, so with this, let us see the course objective. Like we'll see like what is bioinformatics. I have already talked a lot about it. What is bioinformatics? What is the bioinformatics discipline? And the objective of this subject is to make students familiar with the biological questions that can be addressed by bioinformatics tools, and not just familiar with the major biological questions to be addressed by bioinformatics tools. We also take it very seriously that how you can contribute yourself in developing a specific tool. Normally, when we used to do offline classes, I used to give assignments on a regular basis, and I used to ask students to give seminars on a specific topic, and these are very much a possibility, and we will try to arrange some like student projects, student seminars during the course. So that is one aspect how I try to make this course interactive. And there are many areas where you can study much deeper, and you can write maybe a simple code, maybe design a very simple tool to address some of the biological problems. The main objective is here is like, since this is a master's course, you should maybe even if one of you get some insights and get if I can invoke interest in at least even one student, then that would be, I'll consider the course is successful, because this is an advanced topic, and you should always think this as an advanced topic, and how you can actually how you actually see the biological problems, and if you have any unique solution in mind, you should come out with questions, and you are free to discuss with me, and you can write small programs or something. I can tell you, like, I'm teaching this subject since last 5-6 years or even more, and we had even students who published papers while studying this course. So I am very happy to share those things. Like, if you have any interesting suggestions, you can try yourself, and you can write a small. You can try to design a small tool and try to solve a small problem. See, doing research and doing studying advanced courses, it is not always about grades. Okay, so it is about, like, changing your perspective to a specific domain. So if you find a renewed interest in a subject, then the objective of the course is actually successful. Okay, so I think you should take the course in with such an intent, and do not think it as something which can fetch you good marks or something like that. Okay, so that is definitely one objective as a student, but I should say that uh, this these aspects, like going deeper into a specific domain, is also one of the major objectives as a master's level elective course. Master's level is already an advanced stuff, and if you have an elective in a master's course, definitely there is a purpose, because you, in your master's course, you have 50% of your course designed for your research activities. Okay, that is your high-end master's thesis. So when you have a elective course in the second semester of your master's thesis, then it is something which should invoke new queries, not just solve all the bookish problems. Okay, so I just want to tell you that there are ample opportunities in our daily lives when we talk about biology, and when you can connect it to your already acquired computational knowledges. So many things can be done. So to introduce, another objective is to introduce major tools used in sequence structure analysis, explain in general how they work and their limitations, and also, like as I said, it is rightly said that what are the limitations and how you can overcome it. Can we design something unique? If you study this subject, can you design something unique and do a nice work? I had a master student maybe 3, 4, 5 years back, who published during his masters a IEEE transaction paper with me. Okay, so studying a course in biology, bioinformatics, and publishing in IEEE transaction on bioinformatics, computational biology and bioinformatics is quite an achievement. Okay, so during, it's not in his PhD, in her PhD. So she then went abroad and currently doing PhD in National University of Singapore. And see, that is what since this is about course objective, so I consider that as a objective of this course. If I can at least trigger some interest in even one student, then I'll consider this course as a successful one. So that is the objective. And what is bioinformatics? These are all bookish definitions you can read anywhere. And the field of science in which biology, computer science, and information technology merge into a single discipline. So I gave one example of my collaborator from Poland, Warsaw, and where you can see that physics, chemistry, biology, computer science, information technology, everything comes together, and you can actually have very nice interdisciplinary research. And this is all about bioinformatics, a perfect example of interdisciplinary studies. And what is the ultimate goal? The ultimate goal is to enable the discovery of new biological insights. Okay, like we are living in the COVID era, right? So this is the pandemic era, and you can see that how bioinformatics is helping to solve problems. So I gave you an example, like I'm guest editing a special issue where computational tools to combat COVID, and you have a lot of work, computational work that is being done everywhere, and you can see that how people are coming up from many different disciplines, and how you can actually help to solve a complex biological problem, be it COVID or maybe some other disease or maybe something else, even just prediction of the model. Okay, so everything is meaningful. So this is like discovery of, this is the ultimate goal, the discovery of new biological insight, as well as to create a global perspective from which unifying principles in biology can be discerned. So eventually the study is to address new aspects in biology, while the progression of the course, I'll keep giving you many different examples, like my personal research, from my personal research domain to any other stuff that I know. And you should be able to see that this is not just a simple course, or like you are studying operating systems and you are just, okay, operating systems, I know this thing happens, that thing happens, but are you really working with operating systems? Have you ever seen how it works? Do you work with the GNU consortia? So that those things are important, that when you study a subject in your master's, high-level master's course, then you should see like how the course is progressing, why the course is there. So this is the ultimate goal, like when you generate an interest to the subject and you dig deeper and you ask questions to yourself, that how you can utilize your knowledge. Being a student of Jadavpur University, you have seen that Jadavpur is has ranked among one of the top institutes in a recent survey. We all know that it is a great institute, but when you get like some publicly published recommendations or something, then it gives you an extra boost. Otherwise, Jadavpur is often known to the general Indian academia for many different bad reasons. But in general, like Jadavpur's academic excellence is also unmatched, and it has almost ranked among the top 1,000 institutes globally, academic institutes globally. So the rank is somewhere around 1,068 or something in the global rank. So that is a fantastic achievement, considering the limited resources that you get. You are the main like the cream students. So when you go to a top institute, no, if you are in MIT or in a very top institute, you never study the subject just for getting marks. They never do it like that. So you need to get the juice out of it and think about this ultimate goal. What is the objective of this course? And think that way. So that is my objective, is that just to mold you towards realizing the goal of this subject. What is the central paradigm or central dogma of molecular biology? As I have already said in the last class, that, and also showed you some video. I'll share you some more video links. I'll share all the slides with you beforehand, and you should be able to see that how this thing actually happens, like from the chromosomes that unfolds in the form of a DNA strand, and in the DNA strand there are certain regions as genes, and those genes you cannot cut that gene and then take it out. So you just make a copy of that genetic segment, like you photocopy from a library, a very precious book in the library, and then you actually transport that information in the form of messenger RNA to another entity in the cytoplasm, which is called a protein, which manufactures the protein. And this is such a fabulous mechanism, like if you see and if you study this deeper, and it works in every cell, everywhere, continuously, in real time. And this is such a amazing mechanism in biology that it should really fascinate you. Okay, so everything in our life is happening around these three steps, from the gene to messenger RNA to the proteins. And we call it that all the study of the genes is like genome, all the messenger RNA, this is transcriptome, and this is like proteome. The study of this genome is called the genomics, transcriptomics, and proteomics. So this is the objective, and you have in 2010 you have around it is old data anyways, so you can see that there is a huge progression in the decoding the genome of an organism. Okay, so you can see that different types of genomes, different types of organism, eukaryotes, bacteria, and archaea, you can get different types of genomes sequenced. Genome sequencing means that in human, what is the information stored in the chromosome. So you can actually unfold the complete DNA strand, and you can see that what is stored in that DNA, what are the information stored. So for different organisms, you can actually extract that. And there was a 1,000 genome project. So that means you are actually extracting the genomic information of 1,000 different organisms, on 1,000 different organisms. Then we actually expanded that 1,000 genome project to human genome project. That means expanding that to a human genome. And with the easy availability of genome sequencing hardwares, like the technology of genome sequencing, you can actually have many different interesting developments coming up these days. I was telling you about that drug repurposing. So these days there are also studies about customized drugs, right? So when you take a drug, or when you take any medicine, this is a generic medicine, right? That means all of us have different genetic codes somehow, and we really do not know how those codes are encoding proteins in our cell, in our human body. So every human being is different in some form. There are significant similarities, but the genetic sequences are different for every individual. That's why we are different. So when you have a very critical disease, how can we have the same medicine for all individuals? So these days, people often try to, means maybe one day there will be complete genome map, genetic map available for, suppose if I extract my genetic code, and if I store it in some file, and if I fall ill, if I become sick, then the doctor can actually consult my uh, sequence file, and the dose and the molecules that I might need can be designed based on my genetic code. Okay, so in advanced countries, the trend is actually going in that direction, especially these days for the next one, that is the Cancer Genome Project. There are actually situations where in US you have different types of cancer patients, and for many cancer patients, they are actually sequencing the genome. The chromosome is the complete sequence that they get, and some of the drugs they are administering in a personalized basis. So if you can search in Google that what is about personalized medicine, then you should get some information. Those are not in within our scope of study, but I'm just, this is, since this is an introductory class, I'm just sharing that information. So in general, we have 25,000 and even more. So my slides are getting older by every passing day, and you have 25,000 genomes and more. And what next? So we have now, we seem to have sequenced the genome. You have an organism and you have sequenced the genome of the organism. Okay, so that we have already done. Now what next? So we call this as post-genomics era. Post-genomic era. So there was also a misunderstanding. When scientists first started decoding the genome, they thought that if you can decode your genetic sequence, then probably you will be able to solve almost all the mysteries of a living organism, because those genes are the main components that encode a protein, and the proteins are the main entities that drive a living organism. So they're the building blocks. So if you know the programming that generates a protein, that enables generation of a protein, then you will know everything. And you can, if you can do some kind of genetic engineering, then probably you can correct a diseased protein or something like that. But that didn't happen exactly in an expected way. And when scientists actually decoded the whole genome, they realized that there is something even bigger. And there, those studies are actually coming into play, and now people are less interested in decoding the genome. So, okay, we have human genome project, we have 1,000 genome project, we have decoded 25,000 genomes, but just by decoding genome will not solve the problem. So, it is said that we now live in a post-genomics era. Initially, there was a huge thrust in the genomic studies. Now the shift is slowly moving towards the post-genomic segment. The transcriptomics, the proteomics, they are gaining momentum. And there are many stuff that are important in this, like how we annotate there are many things I'll discuss, like what do you mean by comparative genomics, what do you mean by annotations, what do you mean by functional genomics, what do you mean by systems biology. And the main objective is not just decoding the genome, but we need to understand the living cell. Even if you model the activity of one single cell, then you may probably solve a big mystery. And people are still struggling to completely model a single cell in a proper way. And these are like still open problems, how we model a single living cell. There are a lot of researches going on. If time permits, I'll discuss sometime. So modeling of a living cell. So this is the major question, understanding the living cells. What do you mean by annotations? Annotations like these are uh, our genetic annotations. Okay, AC, TGAC, ACTG. So that we'll have to see that these are actually different nucleotides. There are 4 different nucleotides, and there is also a different nucleotide that we get in terms of your messenger RNA. Okay, so that we will discuss briefly. And when we have those nucleotides identified, we marked them with some specific characters. Okay, and when we write a genetic sequence, we mark the genetic sequence in a one-dimensional array of these characters. A, C, 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 T, G, A. So all these are sequential characters. So when you see a genetic pattern, so you will essentially see this code. Now, from gene, we have actually 3 nucleotides group together and form a specific amino acid. So this is a annotation for genes, and we also have annotations for amino acids. So annotations help in identifying the gene within a given sequence of DNA, identify the sites which regulates the gene, and predict the function. This is a very interesting point. Identify the sites which regulate the gene. This is a very interesting pattern, a very interesting insight, that even if you have in your chromosome a gene present, the gene may not be expressed. What do you mean by gene expression? That means you are not getting a protein corresponding to that gene, and there are some regions which are expressing those genes. Those regions are saying, okay, now this gene has to be expressed. That means generate a copy of this gene. But if that gene regulatory segment does not express it, so suppose you have a huge talent, and however your environment, your situation does not allow you to pursue your talent, so that there is no meaning of that talent, right? So same thing happens, that there are gene regulatory segments that regulates the gene. So you can, if you work on those segments, you can silence a gene, you can express a gene. Okay, so many things can be done. So you can do genetic engineering. Suppose you were, I have seen some experiments in the mouse model. I'll tell you those things. And if you want that a gene which is responsible for the skin color of that mouse, if you silence the gene, so the mouse will be apparently albino. That means it will not have a melanin content in its in the mouse. So you can silence a specific gene that produces a skin color of a mouse. You can silence a gene that produces, for example, the hairs in the mouse. Okay, so you can, these are all part of genetic engineering, and you can identify the sites which regulates the gene, and you can play with that. You can also try to predict the function. Interestingly, when you have a complete genomic sequence, not every segment is meaningful, and we do not know why. We do not know why, and there are so many uncertainties, so many unsolved questions in biology that really people do not know why. Why we have such a long, unnecessarily long thread of our chromosome and DNA sequence, and only some parts are encoded into genes. So identifying a gene segment in the DNA sequence that are responsible for a meaningful functionality that we know is a struggle. It is like finding a needle in the haystack. Identifying a gene is a big challenge. So people often, there is a big study like gene finding algorithms. There are often gene finding algorithms. Uh, so there are different signatures for genes. For example, like there is a promoter sequence, open reading frames, etc. And a gene is characterized by several features. Some are easier and some are harder. But there are so much variability. In one hand, the biology is bound by such a rule book based system. Everything appears to be orderly in one hand, and on the other hand you will see that there is so much variability, and the scientists, biologists, they struggle to cope up with the variability, and we do not know why. When there are rules, let there be rules, but it's not like that. And in one side you have very stringent rules and everything work on the basis of rules, and on the other hand you will see that there are so much variability and so much exceptions, so many unknowns, so many free variables are there, and you struggle to find a specific stuff. Okay, so for example, this is just one of many examples that how you can find a gene, a segment that encodes a protein in the genome. Can we identify that? You have seen that there was a Nobel Prize like us in the last class that who identified the gene that defines our body clock. Okay, so that is responsible for the body clock of of a living organism. So if you can identify that gene, means that person identified that gene. So if someone can identify a gene which really drives a specific activity, then this is a big thing. So many unknowns are there. Using bioinformatics approaches for gene hunting, relatively easy for simple organism, but very high complexity and extremely challenging for complex organisms. Not just in humans, shrimps, and even in a mammalian family, they have they're extremely complex. The mammalian family. So you have also comparative genomics. I was talking about annotations, and you can see that when you talk about gene finding, that means maybe this part is responsible for, second line is responsible for a specific gene. Maybe this is the starting of the gene, this is the ending of the gene. So you need to find that. So from a sequence like this, you can find a sequence which is responsible for a gene. So this is like from the annotation, you can apply some gene finding algorithm, and you can mark, okay, this is a candidate for a gene. You can mark that. So you can use bioinformatics for gene hunting. comparative genomics, we will actually study comparative genomics in our, unfortunately, we will not study gene hunting algorithms in our course, but we will definitely study comparative genomics. Comparative genomics is important to know our ancestry. I keep talking about mammalian family, chimpanzee, mouse model experiments. Why mouse model experiments? Why not? Why guinea pig experiments? So all these are because we have significant similarities. We have significant similarities, and we knew about those similarities because of comparative genomics. We studied the genome of chimps, we studied the genome of humans, we studied the genome of mouse, and we knew that they are significantly similar. So comparison between the full draft of human and chimps revealed that they differ only by 1.23%. So humans and chimps are almost 98.7577% similar to each other. Now you need to see the conservation, like where are the similar and where are the different. If you go to the genome, you will see that maybe human, chimps and mouse, they have some similarities somewhere and there are differences somewhere. So there are conservation in some aspect, there are deviations in some aspects. So we need to understand that. So you need to align the sequences to identify that, okay, these regions are more or less conserved and there is a mutation. There is a change from C to G and again G to C in this. So there are certain regions where there is a shift, mutation. Mutation means there is a single point change. And mutations essentially, they are the sole reason for evolution. When we have billions of years of evolutionary history, millions of years of evolutionary history, and you see that from a single cell amoeba, and you actually develop as a complex human being, all because of periodic mutations and your phylogenetic tree. We call this as a tree of life. How they started deviating from one another. Definitely, human is not a chimpanzee. A chimpanzee is not a mouse, but they all belong to the mammalian family, and you will see that in the complete mammalian family, there are a lot of regions where they are significantly similar, and there are some regions where they are differing, and they started to differ, and there they have started to become new organisms. So those 1.23% of difference made human a human and not a chimp. But there are a lot of regions where we are same, and we can actually study those regions, and that's why I would like to focus that we experiment with mouse. For example, for our neuro disorders, and there are a lot of neuroinformatics studies that I also personally engage with a lot of studies where we study a mouse brain for different stress-related disorders, depression-related drugs. And why we study mouse? Because the mouse and the human brains, structure-wise, they are highly conserved. Highly conserved. Okay. So we can actually study a mouse brain and design a drug for a human brain. Okay, definitely there are certain ethical steps and approval steps that you need to take. You need to take up from a mouse to a larger organism, and then only you can apply to a human. But the initial studies, you cannot kill chimpanzee on a random basis, but you are allowed to kill mouse. You are allowed to kill even a smaller, and there are certain regulations, like you go smaller and you get, it becomes easier to get ethical clearances. So even there is a smaller organism called opossum, and these days biological labs, they extensively use opossum because it is even easier to handle, easier to kill, easier to reproduce, and it eats less, reproduces more, so it's cheaper. But again, it is very similar to the mammalian family, and you can study that. So in early days, people used to say, okay, guinea pig experiments. So guinea pigs are big stuff. No, you need to feed that. So it is more expensive, larger animal, and it is difficult to manage. So in the labs, no longer they play with guinea pigs and kill guinea pigs. Okay, definitely there is a progression. So first one organism, then you progress to next level of organism, next level of organism, and so on. So as I said, you can see that comparative genomics says that if you plot the genome of mouse, rat, dog, opossum, chicken, and so on, so maybe these regions are highly conserved. So maybe you can see that, okay, so mammalian family, these four, they are very highly conserved in this region. So if you say, for example, if this is the neuro protein segment or neuro segment, then you will probably see a very high conservation. But if you see something else, then some other regions of the genome which are responsible for maybe some other parts, maybe body parts, then maybe they are highly different. Then you also have functional genomics, as I was telling you, that to be is not enough. You have that gene, but having that gene is not enough. It has to express. The gene has to be functional. What do you mean by functional? You must generate a healthy, functionally active protein out of that gene that is present in your genome or in your chromosome. And that is important. So this is actually yeast genome. This is on a microarray chip. So in general, what we see that we can have a microarray analysis, or we can also call it a gene expression data analysis, where you can see that from different samples, you can see that which genes are expressed and which genes are not expressed. So in this plot, you can see that each row represents different samples, and each column represents one gene. And you can see that greens are responsible, indicates that this particular gene in this particular sample, they are highly expressive. So there are also co-expression. Like, okay, you can see that in this row, this green dot and these two green dots and these two green dots appear simultaneously. So if this is expressed and these two are expressed, then probably this expresses. So there are also co-expression, simultaneous expressions. And this gene expression data gives you a real insight that what you are actually producing as a protein. Okay, so there are some certain regulatory segments in your chromosome that regulates the activity of the gene. Okay, and if it is up, instead of, sometimes it is upregulated or downregulated because of certain conditions, then maybe you had the gene, it used to be producing that particular protein, but for certain reason it may stop producing that. But these are certain cases which are actually having very significant implications. Okay, very significant implications, and that may need genetic encoding or genetic engineering to correct that error. Okay, so gene expression data analysis is a very interesting topic. I usually cover it very briefly. Sometimes very quick discussion is there. So from gene expression data, we can actually see what does the genes do when it is needed, what does other genes or proteins interact with it, and what is going wrong. So what is happening, what is going wrong, so everything we can actually study. Systems biology is also important. We all are familiar with, say, for example, graph theory. Graph theory, you have all studied graph theory, graph theoretic networks. Okay, so you can actually see that the proteins are interacting with each other. Proteins do interact with each other. And you can form a graph of interacting proteins. And this is a biological network of interacting proteins. Okay, so this is part of systems biology. You can, once you can encode this as a graph, you can apply different, say, graph theoretic approaches. For example, graph clustering. You can apply shortest path between two proteins. What is the density of a node? Okay, how the neighborhood analysis. So you can do everything once you represent the network in the form of a graph. Okay, so you have systems biology, and you can actually form biological networks. You can find what can be learned from the biological networks. So you can see that, okay, so there is a hub protein. So this protein is interacting with so many other proteins. So there must be a difference, right? So this protein is interacting with only one protein, but this protein is interacting with so many other proteins. So what is this? Is this a significant protein? Is this an essential protein for the organism? So when you have an essential protein for the organism, then this can be an essential drug target. It is also, unfortunately, a popular target for a virus. Okay, when you have a viral protein or pathogen, any disease, when there are certain you hear me?
Yes, sir. Now we can hear you.
Actually, there was a power cut, so I got disconnected. Can you see the screen?
No, sir.
No, sir.
There was a brief power cut. It got disconnected. Now, do you see the screen?
Yes, sir.
Okay, thank you. So you can actually learn
Sir, not able to hear you. Sir, not able to hear you.
Lovely. নেই স্যার কি পালিয়ে গেল
Hello, can you hear me?
Yes, sir.
Gustav, please mute. Do you see the screen now?
Yes, sir. We can able to see.
Okay. So you can also see that the protein is essential for the organism, or it may be a good target. So then you can also have different types of we'll actually come back to all those things, like we have a chapter on protein interaction networks and different network analysis models, so we'll study those things in details. So now let us come back to more areas like biological databases, and there are many different database is an important aspect of bioinformatics, because this is an interdisciplinary subject and many people have prepared many different databases and they are available in the public domain, and you can actually access different types of biological databases. Maybe it is a sequence database for example, genome browser, like for human genome, if you want to study what is the genomic sequence, you want to, so that you can explore some gene hunting algorithm. It can be a protein sequence analysis algorithm. Okay, so there may be sequence databases. You can also try to explore the structure of the proteins. So there are structural databases. So there are many different types of experimental results. They are also available as different types of databases. Maybe protein interaction network, like in this COVID set, in the COVID situation, people are publishing more and more data. They are experimenting certain variants, though all those things, and they're releasing that information in the public domain. So there are a lot of new databases coming up every month. Okay, so all these databases are somehow connected and logically connected to each other, and you can see that how you can extract some information from, how you can connect, say, a protein connected to a gene, the gene is connected to the organism. So all these things are interconnected. I'll show you some examples. So sequence databases like gene databases, genome databases, disease-related mutation databases, all these things are available. As I said, like there is a genome browser, you can try this UCSC genome browser where you can find all the sequence genomes like 1,000 genome, human genome. So all these genome sequences are available in the genome browser. So disease-related databases, you can see that, for example, uh, for different disease like anemia, blood cancer, you can also have cancer-related databases. In case of sickle cell anemia, we'll come back to this particular disease even in the next lecture. But just for an example, I'm showing you that in hemoglobin, there is a single mutation in this protein in the healthy. This is the image of a healthy hemoglobin, and this is the image of a sickle cell anemia. So these are not healthy or diseased hemoglobin. So there is only one swap from A to T causing amino acid to be valine instead of glutamine. So if you see that this is the genomic sequence, genetic sequence of a healthy hemoglobin in a healthy individual. So it is GAG. This GAG encodes glutamine. Okay. So this is E is glutamine. But when this A, there is a mutation and it becomes T. A becomes T, there is a mutation and this nucleotide changes and the corresponding amino acid formed is valine instead of glutamine. And this results in sickle cell anemia, blood cancer. I was working with one of the doctors in Kolkata Medical College last year. So who was also telling like sickle cell anemia is a big problem mainly in Orissa, Bengal, Balasore, Mednipur, this region. Sickle cell anemia, there are certain genetic traits that actually result in many sickle cell anemia patients in these regions. It's a very deadly disease, and especially for the teenage child, and it's a very painful disease. Not much of you can only do blood transfusion, and there are some medicines, but they are not really effective. So these are unfortunate situation, and then you have some more information like structured databases, like you have 3D structure of proteins, nucleic acid, molecular complexes, and there are many mechanisms how we can actually extract the three-dimensional structures, like mainly X-ray crystallographic experiments, NMR and X-ray crystallographic experiments are used for 3D structure of proteins. So for example, this is a three-dimensional structure of protein, and this is the double helical genetic information, and you can have a club with that is a protein. So we'll come back to how the protein structures look like, why they are like helical shapes here, what do you mean by these wide shapes here. So we'll come back to those segments later. But in general, there are different types of, right now we are discussing databases. So there are different types of databases of experimental results, for example, microarray data analysis, proteomic expression data, then metabolic pathways, protein-protein interaction data, regulatory network, and lot more. There are literature databases. People publish results, right? So you observe certain findings. For example, in COVID, people are observing some results in their lab and they are reporting it to a literature. For example, in a Nature journal or in a Transaction or in a Cell journal. And when that data gets published, then in the database you mark that, okay, this result has been published by this person. So for every journal publication there is a PubMed ID. Medical publications are given a PubMed ID, and that PubMed ID is associated with that result. So you can find that what are the which people are responsible for this result, who invented this and who worked on that. Putting it all together, so you can see that each database contains specific information and link to other biological systems, and these databases are basically interrelated. And you can see, just this is the last slide, I can show you that you have a protein related database, disease related database, genomic data, then many other pathways, structures, gene expressions, literature, and many popular, like for example, Swiss Prot is a very popular protein database. KEGG is a very popular pathway. So for example, PDB is a very popular protein structure database. Okay, so PubMed is the literature database, and all these are connected to each other. So altogether, this forms the domain of bioinformatics. You can study anything. You can focus more on, say, genomic data. People study on proteomic data, disease-related data, gene expression data, and so on. So with this, I end the presentation, and let me stop the recording. I don't know what happened with the recording. I got. Since I got disconnected twice, I'm not sure how the recording will come, but in general, it should come smoothly. Anyway, so I just gave you a brief overview of different aspects of bioinformatics, starting from comparative genomics to computational biology and all these things. So from the next class, we will take up one stuff, mainly we give a great emphasis on sequence analysis. In the sequence analysis, I'll discuss different types of sequence analysis methods, and all these things will be discussed. So if you have any quick questions, you can ask me.
Sir,
Well,
I just asked that I want to ask what is the difference between gene and genome?
genome is like the complete organism is the genome. Okay. And gene is responsible for one particular protein. Like in a complete genomic sequence, there's a long sequence. So many chromosomes in many different organisms. You have different pairs of different sets of chromosomes. And if you unfold that sequence, they are usually packed tightly together. Okay. It's a long sequence. This is a complete long DNA strand. Okay, and in the DNA strand, maybe a small fragment is a small gene, and that gene is responsible for some very specific activity. Say, for example, a particular gene is responsible for generating the protein, say, for example, insulin. Okay, there is a gene which generates the protein insulin. So this is a small gene, and in human genome, and there are many chromosomes, many strands, so many genes are there. Got my point? The
Yes.
complete collection of the genes is called a genome in any organism, not just in human. We call it human genome. 1000 genome means for 1000 different organisms we have sequenced the complete DNA strand. So sequenced means we have extracted the sequence of the DNA, complete DNA. And in the DNA, we really do not know all the time which part is a gene and which part is redundant. Unfortunately, that's what I was telling. We really do not know why there are so much redundancy. There are a lot of redundancy in a DNA sequence. Only a small part is responsible act as a gene that during the process of transcription that gets copied and comes out of the nucleus and goes for translation into a protein. But what about the rest? The rest regions are mostly unknown. Some parts are definitely gene regulatory networks that regulates the activity of the gene. Some parts are redundant. People really do not know what is the purpose. Even in a protein, there are not all the parts are relevant. There are a lot of redundant regions even in a protein. During splicing, when there is a messenger RNA gets formed, during the splicing, you cut off many segments, and some parts are only taken. Why you cut? And there are different types of splicing and different variations. So that generates many different types of protein. There was the same gene. Because of the variation in the cutting process, you actually produce a different protein. The gene coding was the same, but the splicing was different. So splicing process takes you completely different proteins. Protein was same, but after protein, post-translational modification generated completely different types of proteins. So all these things take place. So altogether, a genome in any organism is the collection of complete DNA sequence available in all the chromosomes in the particular organism. But in that strand, if you have a small segment, then that is called a gene. Nucleotides. So this is a sequence of nucleotides, A, C, T, and G.
Okay, sir. Thank you.
Any other query?
স্যার স্যার আমরা যখন কোনো জিন থেকে লাইক ফাইন্ড করছি মানে ওই জিন সিকোয়েন্স থেকে, তো তার পজিশনটা কি ইম্পর্ট্যান্ট? লাইক মানে আমি কোনো একটা স্পিসিসের সাপোজ 10 নম্বর ইন্ডেক্স থেকে একটা জিনের ম্যাচ পেলাম, আর ওই জিনেরই ম্যাচ পেলাম সাপোজ 20 নম্বর কোনো ইন্ডেক্স থেকে, তো দুটো কি একই হবে?
মানে
আই মিন আমি ম্যাচ পেলাম, মানে জিনটা ম্যাচ পেয়েছি কোনো একটা জিনের সিকোয়েন্স থেকে, ওর পজিশনটা কি মানে ডিপেন্ড করবে?
ইম্পর্টেন্ট হতে পারে, নাও হতে পারে, কিন্তু মানে অনেক সময় ধরো কিরকম হয় যে দ্যার ইজ আ শিফট, হ্যাঁ, শিফটিং এর জন্য হতে পারে। ইউজুয়ালি প্যাটার্নটাই লোকে বেশি দেখে, ঠিক আছে, যে সিকোয়েন্স, কারণ শিফটিং বা কিছুটা শিফট হয়ে যাওয়াটা ইন্ডেক্সটা অনেক সময় খুব ইম্পর্টেন্ট না, ঠিক আছে। তবে এটা এইভাবে এক কথায় বলা সম্ভব নয়। যদি তোমার কোনো স্পেসিফিক কোনো কোয়েরি থাকে তাহলে আমরা সেটা ডিসকাস করতে পারি, কিন্তু যখন ওই জিন ফাইন্ডিং এর হয় তখন ওই একটা স্টার্টিং একটা সিকোয়েন্স আসে, সেই স্টার্টিং সিকোয়েন্সটা যেখানে আসে তখন থেকে ওটাকে জিন বলে ডিক্লেয়ার করতে পারে, ঠিক আছে। মানে একটা অ্যাঙ্কারের মতন একটা জায়গা থাকে। মানে একটা হেডার মতন আইডেন্টিফাইড হয়, সেই হেডারটাকে পেলে তখন আমরা ওটাকে জিন বলে আইডেন্টিফাই করি। অনেক রকম প্যারামিটার দিয়ে যখন যখন জিন ফাইন্ডিং অ্যালগোরিদমস কাজ করে, শুধু কিছু ফিক্সড টেমপ্লেট যেমন সিগনেচার আছে, সিগনেচার গুলো কিরকম সিগনেচার, যে ইউজুয়ালি জিনের শুরুতে কিছু সিকোয়েন্স অফ নিউক্লিওটাইডস এর একটা প্যাটার্ন থাকে, সেটা দেখলে অনেক সময় বোঝা যায়। কখনো জিনের এন্ডে হ্যাঁ একটা সিকোয়েন্স থাকে, সেইটা দেখলে এন্ডিংটা বোঝা যায়। হ্যাঁ, এরকম যেমন থাকে, আবার আরো অন্য বিভিন্ন সিগনেচার দেখে লোকে করে। ইন্ডেক্সিং ইজ অলসো ইম্পর্টেন্ট হতে পারে, যে ধরো একটা প্রমোটার সিকোয়েন্সের এতটা পরে একটা স্টার্টিং প্যাটার্নটা এলো, এরকম ধরনের অনেক ব্যাপার স্যাপার থাকে। ঠিক আছে,
ওকে স্যার।
এনিথিং এলস? If you do not have any other questions, you can I'll share the slides with you. ঠিক আছে, আমি ওই হোয়াটসঅ্যাপ গ্রুপে আমি স্লাইড গুলো শেয়ার করে দেবো। স্লাইডটা অ্যাকচুয়ালি we have the slide in Google Drive, so I'll share the link from the drive. In the first slide, if you see, there are certain videos. There are 4 video links. I'll request you to look at each individual video, try to listen to the video demonstrations, and if you have any query even from the video, you can come back and ask me. Sometimes in the class also I used to show the video, but to be honest, you have tight schedule, so I'm not wasting time in showing all the videos. Like 4 videos are there, all the videos are really interesting. You can watch it yourself. I think most of them are self-explanatory, and you should get some idea, especially for those who have not studied biology extensively like me. You can study that, and you should get some idea why it is happening. What do you mean by that? এই যে ট্রান্সলেশন, ট্রান্সক্রিপশন, এই প্রসেসগুলো সম্পর্কে একটু বেটার আইডিয়া পাবে। You'll get a slightly better idea about the process of transcription. এই যে জিনোম যে প্রশ্ন করলো, যে আরেকজন যে প্রশ্ন করলো, the question is about how to mark or how to identify the starting point of a gene. Okay, is it dependent on the index or some pattern? Usually it is based on a pattern, but it may be sometimes depend, because there is no start as such. It's a long strand, continuous strand, so indexing is only relative. It's only a relative index, so it's difficult to have an index based search. Usually it is pattern based search. In some cases, like when you do sequence alignment, you do definitely index based, because you know this is the starting point, this is the ending point. But in gene finding algorithms, you have a DNA strand which is really long DNA strand. There the pattern is more important rather than that. Okay, so if you have any quick question, you can ask me. Otherwise, thank you very much, and we will again meet next week. I usually prefer using the same link again and again, so I'll share you again, but just join through this same link. Okay, so I will not send fresh links again and again. Okay. Thank you.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.